Walaikum assalam, sir.
Sir, can we start?
Walaikum assalam.
Sir, can Sir, can we start?
Walaikum assalam.
Sir, can we start? Can we? Sir, can we start?
We need to also request everyone to kindly mute their mic so we can have uninterrupted proceeding, inshallah.
I request everyone to kindly mute their mics. I request all participants kindly mute their mics. Sir, can we start?
Please go ahead.
Bismillahirrahmanirrahim. I welcome you all to the inauguration ceremony of the online certificate course on the diagnosis of COVID-19, sample collection, RNA extraction, PCR, and data interpretation. I request Qari Hafiz Muhammad Bilal for the recitation of few verses of the Holy Quran. On the diagnosis of COVID-19, sample collection, RNA extraction, PCR, and data interpretation. I request Qari Hafiz Muhammad Bilal for the recitation of few verses of the Holy Quran. On the diagnosis of COVID-19, sample collection, RNA extraction, PCR, and data interpretation. I request Qari Hafiz Muhammad Bilal for the recitation of few verses of the Holy Quran. On the diagnosis of COVID-19, sample collection, RNA extraction, PCR, and data interpretation. I request Qari Hafiz Muhammad Bilal for the recitation of few verses of the Holy Quran.
والأرض وضعها للأنام فيها فاكهة والنخل ذات الأكمام والحب ذو العصف والريحان فبأي آلاء ربكما تكذبان صدق الله العظيم.
Jazakallah. As we all know that the ongoing COVID-19 pandemic caused by severe acute respiratory syndrome coronavirus 2 has brought a state of emergency worldwide. Due to this pandemic, which is affected actually the whole world, and underdeveloped countries has been very much affected because of the poor healthcare system. So that, for this reason, COMSTEC has decided to conduct a workshop which is actually based on the practical sessions to see how this test is conducted. The current gold standard test for the detection of SARS-CoV-2 is the real-time reverse transcriptase polymerase chain reaction, real-time RT-PCR assay. In this context, COMSTEC, in collaboration with the International Center for Chemical and Biological Sciences, is organizing a training course on the diagnostic workflow of the SARS-CoV-2 virus. The program includes experimental and discussion sessions on sample collection, virus RNA extraction, and technicalities for accurate diagnosis of SARS-CoV-2, real-time PCR, and data interpretation. Now we have French translation.
Je vous souhaite tous la bienvenue à la cérémonie inaugurale du cours en ligne sur le diagnostic de la COVID-19, collecte de données, l'extraction de l'ARN, PCR et l'interprétation des données. Comme vous le savez tous, la maladie à coronavirus est en train de s'expandre au niveau mondial et a causé un syndrome aigu respiratoire au coronavirus D, SARS-CoV-2, et a reporté un statut de l'émergence à l'échelle mondiale. Le statut actuel pour le test de détection du coronavirus est une polymérase à transcription reverse de la chaîne de réaction test. Dans ce contexte, COMSTEC, en collaboration avec le Centre international des sciences chimiques et biologiques, est en train d'organiser une cours de formation sur le diagnostic de la maladie à COVID-19. Le programme inclut les discussions et la partie pratique sur la collecte de données et sur l'extraction de l'ARN viral et les facilités techniques sur le diagnostic de coronavirus 19, PCR et l'interprétation des données.
Thank you so much. Now I would like to request Coordinator General, COMSTEC, Excellency Professor Dr. Muhammad Iqbal Chaudhry, for the welcome address.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Good morning and good afternoon, ladies and gentlemen, boys and girls. I'm just so pleased to see increasing number of participants, and I've been told by my colleagues, Ms. Hanifa Baik and Dr. Atiyat Al-Wahab, that over 400 participants have actually registered for this immensely successful training course. You can imagine that in the world where physical meetings are so difficult now, we are now together for a very important course online. And hopefully soon, as the world will change, we would have the opportunity of being together and learning from each other. You see, never in the history of humanity, humanity has faced problem of this nature. This is a health crisis of unparalleled magnitude, a crisis which has never happened before. Of course, there were many pandemics in the history, but the way this pandemic has spread is unprecedented. Now, if you look into the world, you will find that there were countries which were successful in containing this health crisis, and there were countries who miserably failed to to control this major health crisis or to meet the challenge which health crisis has faced. And if you look into it, you would really find two patterns. Number one, good governance. You would find that countries with good governance were able to handle the situation much better than the others. Uh, Leadership which were caring, leadership which were able to develop good healthcare system, leadership which provided a regimented, coordinated response, were able to solve this problem much earlier than the other. And the second important thing is deployment of science. Science has been deployed faster than ever during this pandemic because science was used as a main tool of taking care of this huge human crisis, and deployment of science has been unprecedented. You see, from uh, from the day when this pandemic has begun to happen and reports start coming, scientists have started working on this virus. And from genome to development of test kit to understanding the virulence, molecular biology, to understand how it is spread, has been absolutely unprecedented. So never in the history of science, history of humanity, science has been so fastly deployed. And if I tell you, there are literally hundreds of thousands of research papers have already appeared in last 8 to 10 months on this pandemic. This is a proof that science has been used. We all understand there's a health crisis, but of course, health crisis of this magnitude eventually lead to social crisis and economic crisis. So this is very important for us to understand that this health crisis has been something which has really led us to think and to think of what the world would look like after pandemic would be over. Perhaps we can think of coming back to normal, but in my opinion, there would be nothing like coming back to normal. It's going to be a next normal, a new normal, because somebody said very rightly, pandemics of this magnitude leave a world afresh, a new one. And this is a gateway between the old world and the new world. So there would be no coming back to normal. In fact, it's going to be a next normal. And we have to prepare ourselves for that. For instance, I can tell you that health will not be a regional or national issue now. It's going to be a global issue. It's going to be global health security issues. Any pandemic, any epidemic, any disease of this unknown nature appears anywhere in the world would be taken as a global problem, because if one region of the world is not safe, all of us are not safe also. So world has to be looking into a fresh, everything has to be looked into the perspective of a global pandemic and global health crisis. Now, COMSTAC, which is an interministerial committee of 57 OIC member states, is actually mandated to look into ways of cooperation between countries, the OIC member states, science and technology cooperation, and to help science and technology innovation and preparedness. Now, science, as we know, is apolitical. It has no religion. It is non-dogmatic. It is universal and it has no boundaries. So it can bring people together. So I see science as a unifying force. It's not only for the OIC member state. In fact, science is for everyone, and anything which we do should really help the humanity at large. So it is a unifying force. It is not only for one religion or one region, it is for everyone. And that is the reason why Comstack has initiated lots of programs. So if you find time, kindly visit Comstack website, comstack.org. Then you'll find many programs such as scholarships in virology and vaccine development. We'll find alliance of virology and vaccine technology. You would find many opportunities where you would really be able to develop your own capacity, and many lectures and events have been organized on virology. So in the next several months, you would be able to benefit from many of these programs. I would invite you to look into those programs and see where you would like to interact and benefit from that, because these programs are specially designed to develop capacity. It is extremely important to develop capacity. We have seen in the OIC region and many developing world that literally no tests were conducted because of a very simple fact that the required capacity to conduct high quality tests were non-existent. There are lots of bright people all over, and it is just a matter of providing them opportunity of learning. And this is exactly what this workshop offers. This is an online training course, and I can see about 284 people already online. And this is an opportunity for you to learn from some of the best people in the field, from an institution which is among the best in the developing world. And you would find that your colleagues at National Institute of Virology and International Center for Chemical and Biological Sciences would be able to tell you how these tests are conducted. We all know that the technology is developed to test COVID-19, but of course, so far, this is the gold standard, as has been mentioned by my colleague, Dr. Atithul Wahab, that PCR-based test is the gold standard, and this is the test which is most reliable. Despite the fact that it's more reliable, the skill of a person how to conduct this test is extremely important, because there are many pitfalls, there are many issues, there are many problems where your deep understanding of the subject and also your deep understanding of how these tests are performed are extremely important. Otherwise, there would be many false negative and false positive. You probably have heard about them also. For instance, banana and papaya is showing positive COVID-19 tests is because a simple fact is that, often there is a lack of skills and capacity deficit. So I hope that this training course will provide you an opportunity of learning. Plus, please do interact with your instructors, with your colleagues in Karachi, ask them questions, and make sure that everything which is presented is properly understood. You already have a manual with you. It provides you lots of information, and I'm sure that your colleagues who are conducting this workshop would be very pleased to provide you further information. But at the end of the day, I think our common mission need to be that we are now with learning towards a mutual objective of developing capacity in the field of virology. Is not COVID-19 alone, but once you understand the technology involved, you would be able to perform many viral related PCR-based tests for other diseases also. With that, I would like to thank all the organizers, my colleagues in University of Karachi, Dr. Rashid, Dr. Sabah, Dr. Hana, Dr. Atia, my colleague here at Comstack, Ms. Hanifa, and all of you for your active cooperation, your active participation, and for the preparation of my colleagues who have actually done a lot of work to make this workshop a successful one. I hope this would benefit everyone and this would benefit humanity at large. Thank you very much on behalf of Comstack. Welcome to this workshop, and I hope that in future also we'll get opportunities of learning together. Thank you.
Thank you so much, sir. Now I would like to request my colleague, Dr. Kasali, for the French translation of the welcome address.
Le professeur Mohamed Iqbal Choudry, directeur de l'iccbs, vient de s'adresser en ces termes. Donc, il évoque les problèmes des coronavirus, que c'est un problème à l'échelle mondiale. La rapidité et l'expansion de la maladie Il a évoqué la difficulté à tous les pays, y compris les pays développés, les pays en voie de développement, de pouvoir répondre à ces problèmes. Donc, les problèmes qui est lié à la difficulté de pouvoir contrôler cette maladie immédiatement. Et il a ensuite évoqué la nécessité de pouvoir résoudre ces problèmes rapidement, vu le nombre de morts causés par cette pandémie. Dans son deuxième adresse, il a survolé, il a insisté le fait de pouvoir comprendre la pathologie, la maladie, la nécessité de pouvoir bien la gérer, puisque maintenant c'est un problème entre les nouveaux, il a situé le problème entre les nouveaux et l'ancien monde. Donc il y a la nécessité de pouvoir assurer une sécurité globale sanitaire. Donc c'est un problème sanitaire, il faut des réponses globales, notamment avec les perspectives pour la prise en charge globale de cette pathologie. Et pour cette fin, il a insisté sur les rôles que doit apporter la technologie dans la prise en charge de cette pathologie. Et il a fait aussi savoir les opportunités d'apprendre à travers cette session de formation. C'est une grande opportunité pour pouvoir apprendre sur les méthodes de diagnostic, les méthodes d'extraction et l'interprétation des données liées à cette pandémie. C'est aussi une occasion pour une meilleure compréhension de cette pathologie, spécialement au niveau de son diagnostic. Il a aussi fait savoir qu'il y a une grande nécessité de capacité pour pouvoir répondre rapidement. Enfin, il a remercié tous les collègues de l'iccbs, Université de Karachi, ainsi que tous les autres participants pour la collaboration active, et il remercie tout le monde pour la participation.
Thank you so much. Now I would like to request the resource persons of the training course to introduce themselves.
So, hello everyone, and assalamu alaikum and good morning. This is Dr. Rashid. A brief introduction. Actually, I have done my PhD from Germany in molecular virology, and then I have served in University of Veterinary and Animal Sciences in Pakistan in Lahore for a few months. And then I joined iccbs in the National Institute of Virology, where I have been working for more than 2 years. So here I have been working with dengue viral research and some of the waterborne gastroenteritis viruses, along with this COVID-19 diagnostic lab. After this emergency situation, our institute has developed this emergency lab for COVID-19 diagnosis, and I am a part of this COVID-19 diagnostic.
Assalamu alaikum. I'm Dr. Sabah Farooq, and I'm working as an assistant professor at National Institute of Virology, iccbs. I did my PhD in molecular medicine from iccbs and my postdoctorate in virology from Germany, Mediagnost. I'm involved in different types of projects such as antiviral activities, epidemiological studies, and genotyping and diagnostics of hepatitis A, B, C, E, and SARS-CoV-2. And I'm also associated with the COVID-19 laboratory at iccbs. I will be with you on the second day of this course, and I hope and wish that this will be a very nice learning experience for you all. Thank you.
Good morning, this is Irfan Khan. I have a PhD in molecular medicine with specialization in cell biology I'm working as assistant professor at the Institute in Molecular Medicine. I have a training in regenerative medicine from USA, the Auckland University, a postdoc, and then I have another postdoc from University of Bordeaux, France, in tissue engineering. I'm working mainly on cell biology and molecular biology of the cells. So currently in this COVID situation, I'm associated with the COVID lab for the diagnosis of COVID-19. So currently I'm working here in the COVID lab.
Irfan is a very bright young man. I'm sorry I forgot to mention his name, but he is an extremely important member of the family, and he has done lots of work for the National Institute of Virology and for the public at large.
Assalamu alaikum and good morning and good evening. I'm Dr. Hana Iqbal from iccbs. I have done my PhD in molecular medicine from PCMD iccbs. I have also worked as a postdoc at BGI and BIG, China, and also at MAM Tubitak in Turkey. My current research focus is towards metagenomics and epidemiological studies of SARS-CoV-2, and also I'm also inclined towards animal disease models. I am currently associated with COVID-19 Diagnostic Laboratory at iccbs. I will be facilitating you to understand the real-time PCR for the diagnosis of COVID-19 and its technicalities along with Dr. Irfan on day 3. I hope to have a really nice and wonderful experience with you. Thank you.
With this, now the 3 days extensive training course is formally started, and we hope that this course will be a nice learning experience for you.
Je vous remercie tous pour les 3 jours de cours intensifs. Il vient formellement de commencer. Nous souhaitons, nous espérons que cette expérience sera une grande expérience pour l'apprentissage pour vous.
So now this is the end of inauguration and the formal training course is going to start.
So, hello everyone again. Again, this is Dr. Rashid. So, I will be with you today, the first part of the training session. So, today we will be learning about the first step of the COVID-19 diagnosis, which is selection and collection of samples. As you all know that COVID-19 is an emerging disease which has raised many concerns in the whole world, and not only it's new in its emergence, but it's also new for the diagnosis because the third world countries has never experienced a acute respiratory infection of this severity. So today in the first part, now we will try to summarize the selection of appropriate sample for the diagnosis, and also how an appropriate sample can be collected. So first of all, the risk assessment for the diagnosis of COVID-19. As clearly mentioned by Dr. Iqbal as well, that the COVID-19 disease has shocked the world, and it has actually reunited many nations as it's a global challenge. The first time the world has realized that the health issues of this nature is not a local issue. Because of its atypical type of transmission and its mysterious contingency, this virus is highly risky also for the medical and diagnostic professionals. What are the risks associated with the diagnosis? Here, it's in highly contagious respiratory viruses. As you know that most of the viral infectious diseases, when they are persistent in a region, normally the transmission is slow and local because of its route of transmission. Respiratory viruses are generally very contagious because the route of transmission is normally airborne droplets. And beside having a respiratory infection, moreover, COVID-19 has proved itself as an edge on transmission over other respiratory viruses. As I am showing you in the slide that the typical respiratory virus, which is influenza, has a transmission rate of approximately 1.2 to 1.6, which actually means that if a person has a seasonal influenza virus, it may infect one or two more people. But COVID-19 has shown even more transmissibility, that one infected individual can infect two to three people by transmitting through the airborne droplets. Plus, it's an acute viral infection, acute of severe nature. Very often, it's mild to moderate, but sometimes it turns into many complications and become a severe disease. Plus, there is no specific treatment options available. Antiviral treatments are not specific and they are reserved for very complicated cases. Moreover, up till now, there are many vaccines which are in pipeline, but there is no vaccine available for general population. These are the risk factors involving handling with COVID samples and dealing with COVID-19 infected patients. How can one protect himself from possible transmission? So this is a second big challenge. But before moving to this slide, I would like to ask my colleague for a brief translation of this content.
La première session de la conférence. La première session de la conférence a porté sur la présélection des données. Donc, il s'agit dans cette partie de résumer comment il faut collecter les échantillons appropriés et comment collecter les échantillons de manière appropriée. Alors, il y a eu des risques pour l'assessment des diagnostics. Notamment, il a été évoqué ici une grande probabilité de contamination. Il y a la possibilité de pouvoir les différentes voies de contamination de l'infection virale, donc les différentes routes pour la contamination. Il a été évoqué comme problème qu'il n'y avait pas de traitement spécifique à l'heure actuelle pour la prise en charge de cette pathologie et il n'y a pas aussi la disponibilité du vaccin.
Yes. So there are different kind of masks available in the market, which I think all have seen. A mask of different kinds. A very popular one is N95. So this is a part of PPEs, personal protective equipments, and for dealing with COVID-19 patient or COVID-19 sample, it is recommended by WHO and CDC to use a mask which can protect at least 95% of the airborne droplets. The pore size for this mask is 0.3 micron, and the hindrance or the trap percentage is at least 94% or 95%. So there are different kind of masks which are technically known as respirators. And there are different kind of respirators. They are different because of different regulations. There are different standards which define their use. Here I am summarizing a list. FFP1 and this is at least 80% that it can actually blocks or halts 80% of the particles. When FFP2 or in short it is also said P2 mask, which is equivalent to N95 and it has 94% blockade. And the most popular one, N95, which blocks 95% of the particles. And N99 or FFP3 is the superior mask. It actually blocks 99% of the particles. And the most superior one is P3 or N100, which actually gives you maximum protection against the droplets or the particles, around 99.95 or 99.97%. So these are the masks which are used normally for dealing with COVID-19 suspected patient or for dealing with the COVID-19 samples in the BSL-3 laboratory. But there are some masks which are used for other purposes, for general use, like surgical mask. Surgical mask also gives you protection, and when we are having no risk of airborne droplets, we are in an environment of without airborne droplets, the surgical mask can be used. For sample collection, there are two kind of PPEs recommended based on in what system we are collecting the sample. So one of the sampling collection strategy is collecting sample through a physical barrier or sample collection in a kiosk or in a container. Here I am showing you a picture. This is our container, and in the later phase I will also show you a video demonstration. In this setting where you have a containment area or a physical container to protect yourself from direct contact with the patient, you need minimal PPEs. The minimal PPEs include an inner gown, normal surgical mask and gloves and head covers, etc. But in normal cases, in clinical setting, when you are dealing with the COVID-19 patients for sample collection or for the treatment, where you have direct contact with the patient, it requires full set of PPEs, including a good mask, at least N95 mask, and Tyvek suit, along with proper face shield and proper protections of gloves and head covers, etc. The world has seen many different kind of problems during this pandemic, and there are quite many learnings. One of the learning is a rational use of PPE. And since this is first time that the world has faced an atypical kind of sample, generally our labs are not equipped with handling with this kind of highly infectious sample, and the form of sample is also atypical, and therefore the equipment, the personal protection equipments were not available initially, and the high demand of these equipments has created a situation where the prices of all these PPEs goes much higher, and it was very difficult for the poor economies to manage this kind of PPEs. And it actually brings everyone to figure out how PPEs can be carefully used. And so there are quite many strategies which can minimize the use of PPEs. For example, the mask N95 can be rightly used where it is required. For example, in the BSL3 area where the droplets can be produced or handling with the samples for sample sorting and sample processing. Similarly, in the clinical setting where we are having contact with COVID-19 patients, and in the rest of the areas where there is no physical contact with COVID-19 patients or there is no risk of airborne droplets, a normal surgical mask can protect. Moreover, N95 masks were considered disposable, and once they are used, they cannot be reused initially. But later on, we learned that N95 mask can be carefully used, and more than one time it is possible to reuse. Uh, how can we make the system to accommodate all of these limitations? It actually there are different things which determines the reuse of N95 mask. For example, the how the mask is used on your face, it should be comfortably fit, not over tight. And then the filtration performance can be checked by breathing air and out. And then we should check for any dent or damage and soiling on the mask. And the upper surface of the mask can also be decontaminated using different decontamination reagents. Then there are different the strategies, for example, ultraviolet germicidal irradiation. We can put in the UV light for some time, or different moist heat procedures can be adopted for the decontamination of your PPEs. Types of specimen. So our laboratories, general diagnostic laboratories deal with blood samples very often. Some of the atypical samples include the stool and urine samples, but this type of nasal swabs, especially in the third world countries, were never a popular type of sample. And this was initially a problem to fulfill all the supplies of all the material which is required for this kind of sampling. The appropriate sample for COVID-19 patient is respiratory secretions. As I said, that the virus reside in the respiratory tract. Normally, it infect the upper respiratory tract in the first phase, where the virus replicate and initially colonize, and then it goes further down to the lungs. So the ideal sampling for COVID-19 is the upper respiratory secretions, which include nasopharyngeal or oropharyngeal swabs. In some of the cases where a patient is very complicated, or it has problems with the breathing, or it is on the ventilator, the lower respiratory specimens like sputum or endotracheal spirits can also be checked for COVID-19 presence. Sample collection. So there are some steps for sample collection. The first step is this patient information. There is a WHO defined criteria how we can screen for suspected COVID-19 patients. As we all know that the symptoms of COVID-19 is very similar to the normal flu or normal respiratory infections like pneumonia, bacterial infections, or other viral infections. And only symptoms cannot determine the real cause of the infection. Here, the interviewer or the person who is dealing with the patient should have a questionnaire who ask and write down some of the patient information. This information is also useful for later use. For example, the symptomology and possibility of any physical contact with the travelers in recent past, or any physical contact with the COVID-19 patient, or any exposure to the highly contagious area like hospitals or diagnostic labs. And this type of information can give you the more precise information about the patient and the possibility for the screening. Uh, Type of specimen, as I said, it's nasopharyngeal or oropharyngeal, and this kind of samples require special training for the person who collect the sample. Inappropriate sample collection may also mislead at the later stage, and can also cause a false positive or false negative result. Type of swabs. So in the later slides, I will also talk about the type of swabs. There are different type of swabs available for the sampling, and in later we will see that what kind of swab we should use. A type of VTMs, viral transport medium, and this term is used for a solution which actually maintains the viability of a viral particle. And this VTM is required also for the COVID-19 samples when it is taken from the patient. And then we will briefly talk about some differences and importance of different options available. Okay, type of viral transport. So, as I said, viral transport medium has a maximum viability of the virus particle. It has required for maintaining the viability of virus. The popular one is growth and okay, so there was some noise, so I was just filtering the noise. Yeah, so beside the nutrients, it also needs some antifungal and antibiotics and some of the buffers to maintain its pH. So VTM is an ideal preparation for taking the swabs, but VTM is generally required for maintaining the viability when you need samples for a long time. For example, you can use this sample and then freeze it and -80 for later use. This medium will maintain the virus viability. Moreover, the purpose of VTM is also then if you need to culture the virus for later use, for research purpose, for example. So this VTM maintains a live viability of virus, and later, if you want to isolate the clinical isolate from this VTM, this can be done using this VTM. But if this is not the requirement, if the only COVID-19 test is the only real-time PCR test is a requirement, then this formulation can be minimized to the minimum nutrient level, and it can be minimized to the PBS or normal saline level. So in this recent pandemic, we have also seen that PBS and normal saline were used for sample collection, But the limitation with the PBS and normal saline is that the PBS can maintain the particle for a very short time, and freezing and thawing of a sample which is taken in PBS will distort the virus particle, and the virus particle will not be viable anymore. Similarly, saline has the same limitation of the virus viability, along with the salt interaction with PCR reactions. So not in every PCR reaction, but in some of the PCR kits, this normal saline salt concentration can interact and can influence the PCR polymerases. One of the ideal preparation which was introduced in the recent pandemic is RNA preservatives. This is a set of solution along with the swabs, which actually has RNA preservative. So there are different type of formulations available in the market. It generally have a lysis buffer which can uncoat the virus. So the virus particle is, as you know, is a particle which is covered with a protein coat along with this embedded long proteins, and this lysis buffer will simply unlock this protein coat and will break up the protein core and will release the RNA from inside. And this RNA then preserved in this preservative. So this is a solution which can actually maintain the RNA viability. So the RNA is preserved for longer duration, even for months at the room temperature. So it does not require a cold storage, and it does not require extensive lysis buffers. And so this kind of vials can be directly used very easily for PCR detection. The second part of the sample collection kit is swab. For the COVID-19 test or for any test which requires a sample from nasopharyngeal region or from oropharyngeal region for the detection of virus particle, a swab must be inert, chemically inert, which actually means that it should not chemically interact with other preparations. For example, for downstream process, there is a real-time PCR. So the material should be chemically inert, which do not influence the polymerase chain reaction or do not influence the polymerases. And generally, this kind of materials are synthetic polymers. Very often, they are polypropylene, polyethylene polymers. These polymers are, number one, synthetic. Number two, have different possibilities of flexibility. So it could be made harder and it could be made more flexible. So the flexibility and hardness can be adjusted based on the type of swab. For example, the nasopharyngeal swab is thin and at the edge it is a bit harder, and on the other side it is a bit flexible because it needs to go inside the nasopharyngeal cavity. Moreover, this material, the swab material, should be non-biological material. Previously, the wooden swabs were used for different kind of swab collection, but since wood or cotton or this kind of material are of biological origin, and these biological materials can definitely interact with the PCR reaction on the downstream process. And PCR reaction is a very sensitive reaction, as you all know, and any biological contaminant can cause false positive or false negative interpretations. And that is why this swab should be very carefully prepared and selected, and also should be this whole preparation should be highly sterilized. So only sterilized preparations, completely clean preparations can be used. Okay. So what patient information we normally record at the time of sampling? So, we record in the patient location, patient name, father name, gender, age, address, national ID card number or any personal identification numbers, his contact number for the possible contact, and the date of sample collection. So this is the personal or the sample information which is required at the time of sampling. Okay, so the specimen, as I said, there are different guidelines set by CDC for the collection and for the accuracy of diagnosis. The nasopharyngeal swab is considered initially an ideal swab for the detection of COVID-19, and oropharyngeal and the second ideal swab. But still, the chances or the detection percentage is approximately 62% in nasopharyngeal swab. Since this COVID-19 virus in the later stage goes in deeper to the respiratory region, and that is why the nasopharyngeal swab is there is a very thin time frame, appropriate time for the collection of swab, and sometimes nasopharyngeal swab, as I said, is not an ideal preparation. And similarly, there are different studies for oropharyngeal swabs which shows that oropharyngeal swabs are comparable to nasopharyngeal, and there are quite many studies which are showing data that nasopharyngeal and oropharyngeal both swabs are not as good as the lower respiratory tract efficiencies. Wait a minute. Look at that. We take a small break here and then start into.
अब ऐसको को इसका डिब्बा लेके आए, ठीक है, इसकी कैटरेज को उधर पड़ा है, आ जाओ पाओ कैटरेज। कितने दीजिए है कैटरेज?
Hello again. So sorry, there was some problem with the screen, which is still a problem not solved. But anyhow, we continue. So this is the information which we need for the record of samples. And as I already told you about the appropriate specimen. Okay, so now sample collection at kiosk. So we will show you the video demonstration how a sample can be collected in a kiosk setting or a container setting. And then I will also show you a video demonstration of sample collection through close contact. And then we will briefly discuss about some of the precautions and some of the errors which are possible. So I'm showing you the video, and once this video will finish, we will have a French translator who will translate the content.
This is the COVID-19 sample collection booth of the National Institute of Virology, iccbs, for suspected patients. The booth has 2 windows for patient dealing. The booth is one of a kind designed to deal with COVID-19 patients with minimum risk and exposures involved. To work inside the container, minimum PPEs are required. This should include a gown, a face mask, and gloves. The patient arrives at window 1, where the necessary information is collected, such as name, ID, symptoms, and travel history. The patient is then requested to move to the sample collection window. The second window is equipped with holes and sleeves. The arms are extended out and the sample is taken. The nasopharyngeal swab is kept in one hand and the vial with VTM is kept in the other. It is important that the patient and the personnel are at the same height and are at comfortable positions. After the sample is collected, the swab is carefully and slowly inserted in the VTM vial, which is then tightly capped. The vial is then handed to the patient and is requested to keep the vial in the refrigerator that is kept outside the containers until it is transported to the RNA extraction lab. The gloves that were used to handle patient and sample are discarded outside, keeping the inside of the container risk-free.
So this is a procedure which shows how safely a sample can be taken through a container. So my colleague will translate this procedure into French.
La vidéo, la vidéo a expliqué comment les patients arrivent au lieu de prélèvement. D'abord, les patients arrivent dans le lieu de prélèvement au laboratoire. Il y a des sas de passage, il y a deux fenêtres différentes et il est recommandé aux laborantins de pouvoir porter les masques, de pouvoir porter les gants et de pouvoir porter les souliers pour la protection, pour la biosécurité. Alors, les patients arrivent. Donc ici, nous avons les laborantins qui attendent l'arrivée du patient. Les patients arrivent. Les informations du patient sont recueillies. Nous sommes à la première fenêtre. Maintenant, il va à la deuxième fenêtre, là où il y a les sacs de passage. Le laborantin prend les récipients pour les prélèvements à travers les sacs des passages pour éviter la contamination. Le laborantin prélève l'échantillon au niveau du nez. Donc, il y a un passage, les sacs de passage pour éviter la contamination entre celui qui prélève avec les patients pour éviter la contamination. Une fois l'échantillon prélevé, il est déposé dans un récipient approprié. Le patient prend son échantillon, il va le déposer dans un réfrigérateur. Une fois le prélèvement, le laborantin enlève les masques, enlève les gants. Et les gants sont directement jetés dans un...
Okay, so this was a demonstration of sample collection and container. And I hope it has shown you some of the insights how the sample can be safely collected. Now I will take a 5 minutes break, and during this break I will sort out the questions which are being asked by the participants, and after this 5 minutes we will have a brief discussion or answering session. So in this 5 minutes I will just work on your questions.
Dr. Rashid, can you hear me? Hello?
Yes, Dr. Hafiz, we can hear you.
Thank you very much for a nice presentation. So my question is, so during the sample collection, so there is any time frame? So one person after giving the sample, there is any need to, before that, in between sample collection, any disinfection is needed to, disinfection is needed to sample area and sample collection area?
Sir, Dr. Rashid will give you the answer of these questions. I noted down. This is Dr. Irfan.
Yes, I know you very well.
Yes, sir. So I noted down. Dr. Rashid is giving you answer for this question.
Okay, okay.
The question, Dr. Rashid will give you one by one all of the answer for this.
Okay,
question.
thank you.
Okay, hello again. So I have got some of the questions. Of course, there are quite many participants, and it's not possible to read even all the questions. I have just sorted some of the important questions. There is one question asking that time frame for sample collection. Yes, this is a very important question. What is an ideal time for sampling? As I said, this COVID-19 virus is a respiratory virus which actually gets inside through the respiratory tract, normally from breathing in, and can then in the first phase can colonize in the upper respiratory tract. So after the exposure, normally after 3 days, nasopharyngeal samples contain a detectable amount of virus. And then on the later, when the virus keep growing inside the respiratory tract, the concentration may increase in the respiratory tract. However, at the same time, the immune responses start reacting. In some of the individuals who actually experience very mild to moderate disease, their immune system is competent enough to clear the virus in the early stage. In those patients who has a very strong immune response, in the first week, there is a possibility that the virus could be cleared up with the immune response, and then after 1 week, you will not find a virus particle in the respiratory tract. But if you have symptoms, that means that the disease is still going on and the virus is replicating inside the respiratory tract, and then this is very unlikely that you do not detect coronavirus inside your nasopharyngeal secretions. Okay, there is one more question. Which media is often used for COVID-19 testing? So, before the COVID-19 surfaced on the world, a normal UTM, universal transport medium, is very routinely used for collecting the virus samples. For example, influenza virus samples. VTM is a term, as I said, it's a term which is used for viral transport medium. This medium is made and used in research laboratories also for the clinical isolation of viruses in the from the clinical specimen. This VTM is also used for virus culturing. Plug assays, a modified form is used. But for COVID-19 test, what is an appropriate medium is UTM, Universal Transport Medium, and what is the best for diagnosis. Here is the difference. So if you talk about only sample collection does not actually explain that what are you going to do with this sample later on. So if you are going to use this sample for clinical isolates or for research purposes, UTM is appropriate, or VTM, any medium which has nutrients, antifungal, antibacterials, buffers in it that can maintain the virus viability for longer duration, that is ideal. But if this is not the requirement, if you are working only for diagnosis and diagnosis for a short period of time, for example, the collection of sample and the detection is within 48 hours, right? Then the sample could be different. Then you have two options. One is which is best one is that RNA preservative. So RNA preservative, as I said, is actually made for this purpose. So it does not have a viable virus because it breaks up the virus code and coat, but it has the RNA, which is used for real-time polymerase chain reaction. So this would be ideal, but again, it's this RNA preservatives are expensive, and if you need a cheaper alternate, the best cheaper alternate would be PPS10, normal phosphate line. This is very easily available, and this is a part of, I think, every lab. And 1 ml of PBS can be used for the collection of nasal swab, and this PBS can be used directly to the PCR reaction. One more question has popped up that this sleeves, as we shown in the media, that there are in the videos that there are 2 sleeves hanging out in the container. So the question is that these sleeves may be contributing for cross contamination, that other patients can get contaminated through these sleeves. So I will just clarify that we actually have a special material. This material is non-absorbent material, and when one person leaves the container, leaves the window, these sleeves are cleaned from outside. There is an ethanol bottle, which you can see in the video, they are staying there. And these sleeves then cleaned by using this ethanol after every sample collecting event. Okay, I think we move on. We're not having much time. So in the later at the end of the presentation, we will again take some questions and we will start some discussion. Okay, so now I will show you the next video. The next video is actually sample collection when you have a live and clinical setting where you are interacting with the patients and you have a patient with the close contact. Yes, and this video will also train you for the collection of nasopharyngeal swab. So this video is made actually to train you how one can collect the nasopharyngeal swab. Because before this pandemic, this type of samples was not popular and even was not known. But we have very limited personnel trained for nasopharyngeal swab collection. And as Dr. Iqbal in the beginning has said that there are many learnings with this pandemic and the world is changing now. So, and viral pandemics are now on the full swing, and we see every 1 and 2nd year there are some viral disease newly emerged. So this training actually aims to train the people for all the necessary techniques to be learned for the preparation of future challenges. So now I will show you.
This video contains instructions for isolating sample when in close contact with the patient. The kit contains a vial with VTM in it. This could be stored at the room temperature. There are two types of swabs that could be used for sample isolation, the oral and the nasal. For this sample isolation, we will use the nasal swab. Complete PPEs are required for this sample isolation. The patient is seated at a comfortable seat and is requested to stay calm. Stand slightly off from the patient to avoid direct contamination in case of sudden cough or sneeze. Tilt the head of the patient with non-dominant hand to avoid jerky movements. The swab is held like a pen and inserted horizontally, touching the floor of the nose. The swab is inserted till it reaches the nasopharyngeal cavity, which is about 2 inches in an adult patient. The swab is then immediately and carefully placed in the VTM vial labeled with patient information and sent to the lab.
So my colleague will translate into French, and then we will talk about this video.
Cette deuxième vidéo porte sur la collecte des données avec la possibilité d'interagir avec les patients puisque le contact est très proche. Donc les données, l'échantillon est prélevé. Nous avons déjà les matériels et les récipients pour la collecte des échantillons. Donc les récipients sont préalablement préparés et vérifier. Alors, vous allez regarder si le laborantin qui collecte, qui va prélever l'échantillon, il est en contact direct avec les patients. Donc, quand vous regardez, il le met dans une position pour les maintenir stables, pour un bon prélèvement d'échantillon. Alors, il introduit le type au niveau de la cavité nasopharyngienne. Donc, c'est un prélèvement profond et parfois même douloureux. Donc, vous allez voir comment il le maintient dans une position stable. Après avoir prélevé, il met l'échantillon dans un tube plutôt approprié. Il ferme hermétiquement. Dans un tube où il y a déjà une solution.
Okay, so we will continue, and later at the end, I will take questions about this video also. The second step is sample processing. The first step of sample processing is sample transport. So there are different procedures for sample transport. For example, if the diagnostic lab is inside the facility or inside the hospital, it actually requires a very shorter transportation, and then the cold storage during the transport may not be required. But if a diagnostic lab on a distant place where the sample collection and diagnostic lab has a long distance in between, then the transportation requires proper cold storage. Plus, the sample must be carefully packed. So according to WHO guidelines, it's a triple packaging protocol, which actually means that the VTM vial should be sealed in a plastic bag. This plastic bag should be a zipper bag, and then this zipper bag can be packed inside another plastic bag and all together in a big bag. So it's a triple packaging, which actually makes sure that if there is any leakage or seepage, it will not cause a contamination of samples or any possibility of getting infection to the handlers. In my next video, I will also show you how we perform these procedures. For example, the sample reception, how we receive samples, what are the protocols for receiving samples, and then how samples can be further processed. And I have already told you about the cold chain maintenance. So in many settings, the samples are transported in an ice box container with the ice cubes or the ice packs inside that can maintain a cooling environment inside and make sure that this coolant may not be out for a very long time. Because as the time goes and this coolants may lose its efficiency. So once we receive the samples, samples must be stored again in a proper storage device. If a short time storage is required, for example, if you want to process the sample within few hours, normal refrigeration at 4 degrees is okay for shorter duration. But after the sample is used, once you have used the samples and you need to store the samples for longer duration, for more than 48 hours, then it must be stored at -20 or -80 degrees. One thing which is very important, that the sample collecting is one event, and then there are continuous chain of events up to the reporting, and there must be a continuation connected chain of communication between this. Otherwise, the sample collection and the reporting may not be correspondent. The samples should be collected in a proper manner, should be labeled in a proper manner. These labels then can be stored in a proper database, and these samples can be tracked properly for all the downstream procedures. Sample processing. So this, in the next video, as you will see, the sample processing actually includes opening of samples. So opening of samples is a risky thing, and it requires biosafety level A2 hood. So when you receive the samples, you put the samples inside the hood, where a person completely equipped with all the PPEs can handle and can check the sample for any possible leakage, and then can remove the samples from the bag and can open the samples. Labeling of sample. So when we receive the samples, the sample package is labeled and every sample is also labeled. And this information must be stored properly. So you need to make a mechanism that every sample can be correctly tracked. And then sorting in batches. So normally, if you have a lot of samples, you have to run samples in a batches. We in our lab can run up to 2,400 samples a day. And we receive hundreds of samples every day, and we run the samples in batches. So if you have a lot of samples, you need to sort the samples into the batches where every batch is trackable and every sample within the batch is also trackable. So you have to figure out the mechanism. How can you efficiently connect all the steps? And then transfer of BSL-3 facility. So once you have prepared all the batches for RNA extraction, this batch can be transported to a biosafety level 3 laboratory for RNA extraction. After this, sample sorting and sample handling, a very important procedure is decontamination of area and waste management. So during this procedure, if you are working in a COVID-19 diagnostic lab where you are handling with hundreds of samples, you will produce a big amount of contagious waste. And this waste is hazardous not only for you, but also for general population. Every waste which is produced should be decontaminated through a proper procedure and then can be processed through a normal waste procedure, which is incineration. So the surfaces where you are working, you should clean before and after. So you can clean the benches with 10% bleach or 70% ethanol before and after you start and end the work. Use of biohazard bags. So every waste must be thrown into the biohazard bags, which then will be autoclaved. And there is a periodic autoclaving of the waste. Every round of sorting or every round of sample handling will end up with the autoclaving procedures. And this autoclaved waste then will be processed on daily basis for incineration. Here I'm now showing you the video of sample reception. So it is starting from the sample reception and ending to the transfer into the BSL-3 laboratory.
The samples are received at the designated sample reception area. The samples are received in complete PPEs. The samples are brought in the cool box, which is temperature controlled. The container surface is disinfected before opening. The samples usually come with a tag and label from the source. The samples are kept at 4 degrees centigrade until processing, which takes place the same day. After samples are received, a sample receiving form is made to keep record. This usually has information of the source, number of samples received, and a confirmation of receiving. The gloves used to handle the samples are discarded before leaving the sample reception area. The samples are transported in cool box to the BSL-2 facility for sorting. The samples are processed in a biosafety class 2 type A2 cabinet. The samples are packed in a biohazard bag with patient information written on it. The sample is checked for leakage. The absorbent inside the bag is carefully observed. The samples are then marked with serial number. The same serial number is marked on the sample packet. This is then given to the personnel making a badge record sheet. This sheet is later used for reporting. The sample packet is then discarded in a biohazard bag for proper disposal. If a sample is observed to be leaked, such as the absorbent inside is soaked with the VTM, it is discarded in the biohazard bag without opening the seal. After the badge is made, it is kept in the pass box, which is equipped with UV light. The samples are collected by the technician working in BSL-3 lab on the other side of the pass box. After the work at the bench is done, it is thoroughly cleaned with 70% ethanol. The biosafety cabinet is also wiped. The biohazard bag used to discard leaked samples is tightly sealed with a tape, making a gooseneck. This bag is then discarded in a secondary biohazard waste bag to avoid leakage. The secondary bag is also tightly taped, making a gooseneck. The bag is then taken to the autoclave. The autoclaved bags are then sent off for incineration. This is the end of the sample sorting procedure.
So this video actually explained the whole process from sample reception, sample transportation into the BSL-2 facility where samples are sorted, and then sample sorting and batch making, and then transportation of samples into the BSL-3 facility, and then cleaning and decontamination of the lab. Now my colleague will translate this content into French.
La vidéo ci-après explique comment l'échantillon est reçu, donc la réception d'échantillon. Donc c'est le transporteur d'échantillon amène l'échantillon dans un bac. Comme précaution, le bac est désinfecté avec une solution hydroalcoolique, avec un désinfectant. Après avoir désinfecté, on essuie la partie supérieure, donc la couvercle. On jette ce qu'on a utilisé pour essuyer dans un récipient pour déchets contaminés. Une fois essuyé, le bac est ouvert et l'échantillon est pris est collecté. Alors, vous allez voir qu'il y a toutes les informations sur l'emballage que contient l'échantillon. Après, l'échantillon est gardé au réfrigérateur. Ici, il s'agit d'une fiche qui contient les informations par rapport à l'échantillon reçu. Donc, c'est lui qui réceptionne les échantillons, enregistre les informations et il signe. Avant de pouvoir être transporté, donc les gants sont jetés dans un récipient pour déchets contaminés avant de pouvoir quitter la salle de prélèvement. Les receveurs d'échantillons aussi jettent les gants qu'ils avaient utilisés, portent de nouveaux gants. L'échantillon est ensuite enlevé du réfrigérateur pour être mis dans un second bac, dans un second récipient. L'échantillon est transporté dans un autre local, plus sécurisé que la salle de réception. Pour la seconde fois encore, les bagues et les récipients qui portent l'échantillon est désinfecté. Et on aussi ensuite sa couvercle. L'échantillon est pris. Il est mis dans une sorte de hôte pour la biosécurité, pour la sécurité. Alors, les informations contenues sur les informations de l'échantillon sont prélevées. De même que l'échantillon lui-même, on vérifie. On met un nouveau numéro de série sur l'échantillon, mais aussi les mêmes numéros de série est mis sur les sachets qui contenaient l'échantillon. L'emballage ayant contenu l'échantillon, les informations de l'emballage ayant contenu l'échantillon sont enregistrées dans une fiche. Donc, toutes les informations sont enregistrées sur une fiche. Vous allez voir les différents numéros. Vous avez les numéros des badges et tout ça. Donc, toutes les informations relatives. Une fois enregistré le numéro, le badge est jeté et l'échantillon est transporté dans une boîte avec les rayonnements UV ultraviolets. Alors, une fois encore, la table est essuyée avec un désinfectant. L'échantillon est encore transporté dans un dispositif, mais on prend la précaution de pouvoir désinfecter les dispositifs qui recueillent l'échantillon. Donc, il est ici avec un désinfectant. Donc, le sachet jaune veut dire que l'échantillon attend que c'est... Donc, tout ce qui est emballé en jaune sera jeté dans un récipient pour les déchets contaminés qui est en jaune. C'est une boîte jaune. Les déchets sont prélevés dans les récipients pour déchets contaminés. Ils sont hermétiquement fermés et puis ils sont amenés dans un autoclave. Donc ils sont autoclavés et par la suite ils seront incinérés. Donc, il faut fermer hermétiquement l'autoclave. Donc, voilà.
Okay, so now, up till now, we have seen what kind of samples are required or recommended for the detection of COVID-19 tests. What are the requirements for the COVID-19 testing, and how the sample will be collected in different settings, how the samples will be processed inside the lab, how the lab should be in components and in composition. So this is my next topic. So I will briefly tell you about an ideal COVID-19 testing lab, what units it requires. And I will also show you a video demonstration of our BSL-2 and BSL-3 laboratories, which we are using for COVID-19 diagnosis. An ideal recommendation, or you can say a standard recommendation for COVID-19 diagnosis is a BSL-3 laboratory for RNA extraction. However, when the pandemic was on peak, the WHO has relaxed the policies, and BSL-2 plus laboratories would also be recommended, or would also be allowed for using the RNA extractions. However, in an ideal situation, a BSL-3 biosafety level 3 laboratory is required for RNA extraction. And a BSL-2 biosafety level 2 unit for sample sorting, which you have already seen in our previous video. A PCR pipetting unit. So real-time PCR is a highly sensitive technique, and if you are using a highly sensitive technique, it is very important to keep the containment areas completely separated, and therefore it is recommended that we should pipette in a unit which is completely separate. So PCR pipetting unit actually involves the pipetting of master mix, pipetting of PCR enzymes, and any other diluent if required. And this pipetting is made inside this PCR pipetting units, and then the PCR vessels will be transported to the next lab, a separate unit for RNA pipetting. The next lab or the next step is RNA addition into the sample vessel. And ideally, an RNA addition lab should be completely separated, where you will add the expected RNA into the sample vessel. And then you should have a PCR machine area. So this is a simple lab where you have your PCR cyclers, where you will run your real-time PCRs. And then you have a separate room where you deal with all hard and soft data for processing. For example, your batch making data, your sample records, and your reporting data will be handled in a separate area, which is confined for all data handling. Now I will show you a video demonstration of our lab. Welcome again. So I am standing at the entrance of our BSL-2 and BSL-3 facility. So just after the entrance, there are some accessory labs. On my right hand side, there are 3 accessory labs which are used for DNA extraction, PCR amplification, and PCR cycling. And on my left hand side, these are the labs which are used for PCR pipetting, RNA extraction, and serological work. So since the voice is not very clear inside the lab, because in our lab we have HVAC system, which is a complete separate system for air in and out, and there is some noise inside the lab. I will again briefly tell you what I have said. So normally a BSL-3 lab or a BSL-3 entrance and sub-lab entrances have a different kind of door, which is a bit heavier than the normal doors. Why it is required? Because you have a pressure regulations in every lab. For example, in a BSL-3 laboratory, you have a negative pressure, and for maintaining these pressures, you have heavy doors. Then just after the entrance, I have showed you two wings on my both hands. And in every wing, there are some accessory labs. On the right-hand wing, I pointed out the area where we have PCR cyclers and the room where we deal with all soft and hard data, reporting room. On the other hand, on the left-hand side, I have shown you or pointed out to you three accessory labs. One is used for storage. The PCR kits and all other stuffs are stored in that lab. Then we have a PCR pipetting area. PCR pipetting area, as I have already told you, this is used for PCR preparations. And then we have an RNA lab where RNA is added into the sample vessel. Now we see what else. Now I am standing at the junction of BSL-3 laboratories. So we have two BSL-3 laboratories. On my left hand side, it's one BSL-3 laboratory which is connected with the BSL-2 sub-lab, and similarly on my right hand side there is another BSL-3 laboratory which is connected to sub-lab BSL-2. So this is a sub-lab BSL-2 level. So in this sub-lab we have an empty room, which is an empty This sub-lab is used for the sample processing. So here normally samples are recorded, so there we maintain the sample record. This is a biosafety board, A2, where samples are open and samples are checked, and then samples are processed for batch making. So, in this clip, I have shown you two units of BSL-3 on my both hand sides, right and left. And every BSL-3 unit has a connected sub-lab, which is of biosafety level 2, and which is used for sample processing like sample sorting, batch making, as you have seen in the earlier video. In the biosafety level 2 lab, I have pointed out to you the safety cabinet of BSC-A2. I have pointed out the autoclave machine. It's a 2-door autoclave. So this is an outer part of this autoclave machine. And we see later on. Here we have an autoclave unit. This autoclave is 2-door autoclave, which is connected with the BSL-3 laboratory. Every waste which is produced inside BSL-3 or in BSL-2 can be autoclaved before waste. This is a pass box. Here samples are transported inside and outside through the transforms within the BSL-3 and BSL-2 laboratories. In this part, I have explained you about autoclave and pass box. This pass box is used for things getting inside the BSL-3 or things getting outside the BSL-3. This transport box is equipped with the UV lamp for surface disinfectant, and it has two doors and they are lockable, and one door is open from one side at once. Process for that. This end of corridor is connected. Samples are transported inside and outside through the transform within the BSL-3 and BSL-2 laboratories. So I am again in the corridor. This end of corridor is connected with another corridor, which is a sample reception area. Outside this lab, we have a separate portion where samples are received. So this is showing a sample reception area. So since I am in a minimal PPE set, I avoided entrance into the sample reception area, and I'm just pointing out the area where samples are received, and you see that these two corridors are completely separated. Now I will show you some of the necessary labs in the BSL-3 laboratory. At my right hand side, we have a BSL-3 laboratory component which is used for PCR pipetting and RNA mixing. This is our PCR pipetting lab. This area is used for PCR reaction preparations, master mix, and enzymes are pipetted in this lab, and then the samples processed to the RNA mixing lab. So this is the first lab for the downstream process for PCR. And this is used for PCR preparations, which means making a PCR vial, diluting the PCR reagents, and pipetting PCR reactions. So the cabinet I have shown you in this video is a PCR cabinet, which is used for performing PCR reactions. This is our RNA mixing lab. The PCR plate after the PCR mixing is transported to the RNA mixing lab, and then RNA is pipetted inside the one. This is RNA lab. The extracted RNA is pipetted inside the reaction vessel, inside the biosafety A2 hood, which is shown. So this is our PCR cycler room. Here we have real-time PCR cyclers where we run the PCRs. Welcome again. Is used for the PCR place stop. Run the PCRs. So in this part, I have shown you a PCR cycler room. So real-time PCR machines are kept here. And this is actually the room where we are sitting at the moment. So in my background, when the camera will be live, you will see the machines in my background. Finally, this is our reporting room. Here, the reports are made and then communicated to the patient. So this is our final lab, as I said, for hard and soft data handling and report preparations. My friends, colleague will translate in brief this video.
Brièvement, nous avons cette vidéo nous représente, donc c'est une vidéo démonstrative sur les labos qui est disponible ici à l'Institut national pour la virologie. Donc il s'agit ici d'un labo de biosécurité de niveau D. Et le labo a différents compartiments, donc différentes salles. Donc, ce qu'il faut retenir de manière générale, il y a 3 grandes salles avec d'autres annexes. Donc, ce qui va nous intéresser sont les 3 grandes salles. Donc, on est en train de nous montrer ici les différentes salles pour les différents matériels et équipements. Alors, ici, nous entrons dans la première salle. Donc, c'est une salle où il y a certains dispositifs pour le travail et l'équipement. Vous allez voir BCL3 plutôt. Donc, il s'agit d'une salle pour le stockage. Vous avez une table de travail. Et puis, vous avez un dispositif de biosécurité pour le prélèvement. Alors, dans cette même salle, vous avez l'autoclave. Donc, l'autoclave est pour pouvoir recevoir certains pour la stérilisation. Donc, qui est utilisé selon les manuels et qui est disponible. Et après l'autoclave, nous avons un sas de passage. Donc, comme nous avons expliqué là dans différentes vidéos, les sas de passage pour collecter les données, pour collecter les échantillons. Ça, c'est la première salle. Donc, à retenir, il y a l'autoclave, il y a les dispositifs comme sas de passage. Nous avons une seconde salle. Ça, c'est la deuxième principale salle. Donc, quelques explications sur les dispositifs des différentes salles sont en train d'être données. Alors maintenant, ici, nous entrons dans la deuxième salle ou bien le deuxième grand laboratoire. Donc, c'est une salle pour la préparation du PCR. Donc, la préparation de la PCR, c'est ici qu'on fait des préparations pour la PCR. Et dans cette salle, vous avez, on va vous montrer plus tard. Nous sommes dans un autre labo. Donc, vous avez la plaque pour les réactions. Donc, ça, c'est la plaque pour la réaction, pour les différentes réactions. Ce qu'on appelle la plaque de la PCR pour réaliser différentes réactions. Nous entrons dans un autre labo. Proprement dit, c'est dans ce labo que vous allez retrouver les PCL.
ये पिक्चर्स है, ये वीडियो तो नहीं है, तो पिक्चर है।
Donc, le labo précédent, c'était un labo pour les PCR avec le labo. Mais maintenant, ici, nous sommes dans le labo, dans la salle de reportage des données. C'est ici que sont traitées les données, les différentes données recueillies.
कोई ट्रांसलेटर ही रख लेते।
Okay, so at the end, we'll talk about disinfection of the work area. So, since this procedure involves extensive transportation and handling of material which may be contaminated with coronavirus particles, and this is very important to regularly clean every surface and all the labs. So there are different kind of disinfectants available in the market. So you can check the disinfectants in the EPA website also for the guidance that EPA approved products are better in the performance. There are few cleanings which housekeeping staff can perform for you, but there are most of the cleanings which you have to perform by yourself if you are working in a COVID-19 diagnostic lab. For example, cleaning biosafety hoods, cleaning working bench tops must be cleaned before and after every start and end of the work. The surface areas where you are working, for example, dealing with hard and soft data, the surface areas must be cleaned. This also include the surface of keyboards, mouse, and all other stuff which you touch regularly. Housekeeping staff can maintain cleaning the floors and the surfaces where you touch very frequently. For example, handles of the doors must be cleaned regularly. So for the floor cleaning, disinfectant must be used. This disinfectant should be safe and also efficient for inactivation of virus particles. So this is all about the COVID-19 diagnosis day 1. In my video demonstration, I have shown you up to the biosafety level 2 areas. I haven't shown you biosafety level 3. So tomorrow in the next session, Dr. Saba will explain you the RNA extraction procedures, which is performed inside the biosafety level 3 area. And then you will see how biosafety level 3 area looks like and how you work inside biosafety level 3. At the end, I want to take 5 minutes that I can actually see your questions, and then we will be open for discussion. And there is one announcement for everyone. Please listen carefully. There is an announcement. Kindly mention your correct names in the comment section for the certificate of attendance. Kindly mention your correct names in the comment section for the certificates. Thank you.
Hello. Hello. Can you hear me? Hello.
Yes, sir. There is a 5 minutes break, and then I will be back with all the. I'm sorting the questions now. I will take your question after 5 minutes.
Okay, okay, okay.
अबे भाई मेरे चुप हो जा। चाय पिएगा? चाय खाना खा ले, चाय पी ले। जाके थोड़ा टीवी देखना कुछ जियो
Hello. Hello.
Yes, sir.
Yes, sir.
So Dr. Sir, thank you for a nice presentation. I have one question. So it is mandatory to have a BSL 3 laboratory to start this COVID testing.
It is not necessary. I mean, it is not compulsory. As I said, the WHO or CDC has relaxed its criteria only because of this emergency situation. As you know that in this pandemic, many third world countries and even the developing world has limited number of BSL-3 laboratories,
Yes,
and
yes.
its extensive load of samples requires more laboratories in every area, and that is why CDC has relaxed its criteria. Although the corona virus is a BSL-3 organism, this is highly contagious virus. But for the diagnosis of corona, BSL-2 plus lab is okay for diagnosis, not for virus culture or virus isolation, only for diagnosis.
Thank you, Dr. Sir.
Welcome. Okay, so there are quite many questions. I will try to answer some of them. So there is a question: in normal saline, a suitable media for a normal testing? This is a question. No, normal saline is not a suitable media for normal testing. As I have said earlier, normal saline is used in a limited resource setting. If the supply of VTM is not good, if you are running short of VTM, if VTM is very expensive and you cannot afford the appropriate media, then normal saline or PPS can be used as an alternate. Okay, there is one more question. Is there any contamination on the refrigerator because many patients touch it? So this is about our video, which I have shown in the container, the container video, where patient has handed over his sample and he put this sample inside the refrigerator. So a participant is asking that because of this, that refrigerator can be contaminated and the other patients can also be contaminated. The thing which is missing in this video is a waiting area, where the patients sit and wait for their turn. And this sitting area has hand sanitizers. So every patient which moves to the window are basically hand sanitized before, and then they go to the area where they give the samples. And similarly, when they leave, they also clean their hands by sanitizer. So this is clear. And the question, does patient is in risk if he is putting, this is I have already answered. Can you please highlight the facts and rumors regarding COVID-19 just to eliminate confusions? Well, I don't know what kind of rumors you are talking about, but COVID-19 exists, as you have already seen. I think every one of us now has experienced some of the people in their contacts, in their close contacts, who has experienced COVID-19. So COVID-19 exists. Okay. Now, if patient height is not equal, what can I do? Okay. So what my colleague Kiran has mentioned is not about the actually the height of a person, rather the height at the time of sampling. So because you know you are drawing a sample from nasopharyngeal swab, so you are entering the swab inside nostrils. You should be in a position where you can look at the nose clearly and you can insert in the right position. If the sample is if the patient is very short, he may use a stool. So we have two stools outside our container where that can be used for people. And we also have a chair, a long chair, where the patient who is who cannot stand, who is very sick, can also sit. But you have to somehow adjust the height in a comfortable position that you can access the nostrils comfortably. Okay. The question in a resource limited setting is not and advisable to use triazole instead of VTM for sample collection? Yes, triazole is normally used as a reagent or as a mean of nucleic acid extraction. Yes, if you are directly starting with this, triazole could be an option for the extraction. But again, triazole is not a media which can maintain the viability of a virus particle. Then, is there any chance of VTM contamination with another microorganisms? Well, yes. So this is not actually contamination. This actually exists, not as a contaminant. So when we are drawing a sample, either it is an oropharyngeal swab or it's a nasopharyngeal swab, you are actually collecting everything which is inside the swab. That could be a bacteria, this could be a virus, or rhinovirus, influenza virus, this could be anything else, or whatever it is. I mean, in the nasal swab, you are having it in the sample. The PCR reaction, which is for COVID-19, is highly specific for COVID-19. So you are not relying on swab, but you are actually relying on your PCR amplification kit, which you have used, and these kits are highly specific. You target only COVID-19 viral targets, which are amplified and then detected. So for example, if you have co-infections, for example, if you have influenza or if you have tuberculosis, whatever you have, you will not see anything except COVID-19. If there is COVID-19, you will see an amplification. If there is not, you will not see anything. Okay. If we use PBS, how long, okay, we can remain viable? As I said, 48 hours. 48 hours, you can actually keep the samples in PBS or in normal salines, and the decaying process is very fast in PBS and normal saline, and that is why after 48 hours, the efficiency will go down. Why the patient, okay, this is I've already answered. At the end, kindly share PPT, okay. We have used PBS, how longer the virus will remain viable. I have already said 48 hours. Is there cell lines available for doing in vitro study? Okay, this is out. Actually, I'm not talking about the virus culturing and virus isolation. So you have lung epithelial lines, for example, there you can cultivate this virus, and you can check the infectability of virus in different cell lines, and then that cell lines you can use. But again, virus culturing is out of the scope, and this lab, this diagnostic lab, is not designed for virus culturing. Okay. Is there cell lines? Okay. What is the broad beef media effect on the virus quality? Well, these are the nutrients which actually provide appropriate viscosity, appropriate medium for virus to be viable. Because when you freeze the vial, if the protein content is low in the medium, this virus particle will clump and break. So the viability will go down. This freezing thawing will break the virus core proteins. Okay, why 70% ethanol? Just because it's surface cleaning, as it's why anything to virus? Well, 70% ethanol is one of the very common thing which we use in our labs as a disinfectant, and that is why I have also shown you 70% ethanol. Otherwise, there are quite many disinfectants. You can check on EPA website. Some of them which we use in our lab is 10%. You can use very strong bleach is not good. So you can adjust the bleach 5 to 10% bleach for surface disinfectants. Plus, you can use hydrogen peroxides as a disinfectant. Sodium hydroxide is a very potent virus inactivator. 1% to 2% hydrogen sodium hydroxide can also be used for inactivating the viruses. Normally, influenza viruses or other respiratory viruses which has envelope and popping out proteins can be inactivated by hypoxide treatment, which is sodium hydroxide treatment. So when we work with viruses, we actually discard viruses in a container which has sodium hydroxide, so it immediately inactivates the virus. What should we do if the sample is leaked? Okay, what should we do if the sample is leaked? So in my video, I have shown you that if you have a leaked sample, you will not open the sample, but since you have the information available on the packet, you can note down the information and you can request resampling. So we normally do this, that we report the sample, the leaked sample, and we request the resampling. Okay, explain difference between upper and lower respiratory specimen, please. Well, upper respiratory tract specimens, as I said, that oropharyngeal swabs which you collect from in the throat. So you actually insert the swab inside the mouth and you touch the upper palate in your throat and you take the swab from there. And nasopharyngeal swab you have seen in our videos from where and how you would collect. Okay, the lower respiratory specimens, as I said, it's not easy and it's not a routine procedure. For example, if a patient is very complicated, he is having severe difficulty in breathing and he is on ventilator, then this sputum and this tracheal aspirates can be taken, and these samples can be taken by medical professionals only. Okay, so medical professionals can take these samples and can be tested for COVID-19. With context to oropharyngeal and nasopharyngeal, where we have more chances of false positive and false negative results, I mean, which method will give more accurate results? Am I clear? If not, then explain you the question. Well, it's a big debate, and you will see different studies. In last few months, a lot of studies has evaluated different specimens for their PCR efficiencies. And one of the study which is very often discussed, I have already shared. I have already talked to you about that. This is 62%. The efficiency of nasopharyngeal swab, the detection efficiency of nasopharyngeal swab is approximately 62%, which is higher compared to oropharyngeal swab. But there are more studies which are comparing, and they found it comparable. But as per recommendations of CDC and WHO, nasopharyngeal swab is an ideal candidate for COVID testing. And you ask me why. Well, the studies have shown their efficiencies, and that is why now since there are different different contradicting results, this is very hard to comment on it, but we are detecting the viral genome, right? The virus is colonized in the respiratory tract. If we are taking sample from oropharyngeal region, this is the region from inside, which is thought from inside, and we take actually the salivary swab from deeper down the throat. Well, this region is not the first choice for two reasons. Number one is that you drink water, you eat food, and this region gets flushing very often. And that is why the concentration of virus may be reduced there. While the nasopharyngeal region is a region where you do not have this kind of frequent flushing. And that is why nasopharyngeal swab is even more appropriate for this sampling. Okay, the question between, I have already answered. This is also again oropharyngeal and nasopharyngeal swabs. Okay, what is the suitable cancer cell line can be used for hospital? Well, A509 cells, it's lungs carcinoma cells, are proper candidate for this. But again, this question is not for diagnosis. The virus culturing is completely different thing. What will the minimum distance in mouth which should be kept to gain oropharyngeal specimen? Well, there is no hard line distance explained, but you should be comfortable and you should be, the patient should be accessible, right? The sample should be accessible. So normally 1.51 feet distance is enough between you and patient to collect the swab. What is the suitable cancer? This is again, I have already answered. Okay. Can the COVID-19 test tell the difference between someone with an active infection and someone who was infected in past? Yes. So PCR, real-time PCR test is actually a diagnosis of active infection. And if the virus is, now see, you have got infection, and when you get infection, your immune system will automatically activate, and your immune system will start fighting against the coronavirus. When you have disease, you have symptoms, your infection is active, the virus is growing, at the same time your immunity is fighting. When you have a specimen, your virus is still present in the secretions, and you detected the virus in the secretions. That actually detect that the virus is not completely cleared, right? The virus is still existing in the respiratory tract, but your immunity has cleared the virus. For example, after few weeks, you have taken nasopharyngeal swab again. Since your immunity has cleared the virus does not exist now, and your nasopharyngeal swab ideally should not have any particle. So you will not find, and that is why the recommended test for the confirmation that you are over, your infection is finished, is recommended as real-time PCR. Real-time PCR determines that the negative real-time PCR determines that now you are safe and you can end your isolation. Well, the antibody test, which is the second option, why it is not suitable? Because, as I said, that when you got infected, your immune system will start fighting. The first antibody which will be made is IgM. IgM will be available, and when you will test for antibody, you will find IgM. Presence of IgM indicate there is an infection, right? And IgG comes up later in the adaptive immune response. But presence of IgG does not actually indicate that infection is over, because IgG has to come after the adaptive immune response, but the fighting is still on. If the virus is completely cleared by the IgG, the PCR should be negative. Presence of IgG inside the secretions determines that the fighting is going on, that IgG is working to clear up the virus. However, it actually confirms that your adaptive immune response is now active, and you are, later for later, if your infection is over and you find an optimum titers of IgG, it actually tells you that your immune system is equipped with the protection. What are the suitable, again, okay. Please answer what this question can be answered during the PCR session. Okay, this is what amplification region nowadays have been targeted for COVID-19 diagnosis through real-time PCR. Kindly tell about those parameter specifications on which basis a patient with seasonal influenza or COVID-19 infected is differentiated through this PCR. Okay, so this is a question my colleague Dr. Irfan will be dealing in detail. Very brief answer is that the PCR targets highly specific target of coronavirus. There are different genes, but Dr. Irfan will tell you in detail on the third day. Okay, there are more questions coming up. I am actually sorting up the questions. Meanwhile, we take a short break. Hello, participants. Hello, participants. I will take more questions. I am working on your questions. I am sorting out. I am working on the questions. Meanwhile, I want to show you the videos of donning and doffing. So please concentrate on the videos. These are very important. Donning and doffing are the procedures for wearing and getting off of the PPEs.
This video contains PPE donning instructions for sample handling. Apply 1 pump of hand sanitizer. Rub hands palm to palm. Rub the right palm over the left and vice versa. Continue to rub both hands together until the hand sanitizer is dry. The next step is donning the isolation gown. Double check the gown for defects. Don the gown by inserting the arms into the sleeves with the opening to back. Tie the neck and waist ties in bows that are easy to release, as this will facilitate easy removal by eliminating the need to struggle with untying knots. Step 3 is donning the N95 respirator. Place the N95 respirator on your face, covering your nose and mouth. Gently mold the wire on the nose to ensure proper fit. Perform negative and positive seal checks. A surgical face mask is used to cover the respirator to avoid direct contamination and ensuring extended use. A secondary coverall is worn, which is double checked for defects. The coverall is worn by inserting the legs first. The arms are inserted into the sleeves and the front opening is zipped. The last part requires donning of the face shield or goggles. If you are wearing goggles, ensure that they are not interfering with the fit of the N95 respirator. At no time should eye protection be readjusted in the patient care area if face respirator. The face shield is removed. Once your face shield has been removed, it is disinfected with 70% ethanol. The surface must remain wet for the appropriate wet time. Place clean face shield with the strap facing down. In the next step, the coverall is removed. The zip is opened and the head cover is pushed back. Doff the gown, folding the outside of the gown tightly inwards into a ball to contain the contaminated site. In the next step, the isolation gown is removed in the similar manner. Tear open the neck and the back tie and roll the gown inwards. Gloves are removed utilizing glove-in-glove technique. Take the dominant hand and pinch the palm of the non-dominant hand to remove the glove. The head cover is removed. The masks are removed by not touching the front part. The masks are carefully disposed of in the discarding bag. The waste produced in the doffing area is autoclaved and sent for incineration for safe disposal. The last pair of gloves is removed and hands are sanitized.
Okay. So, the time is short. We will take more questions. Now, so there is a question asking that what kind of nutrients we can use for making VTM. So, Eagle's growth medium, NEM, which is a very popular medium for culturing cell lines, can be used. There is liquid NEM available, but for preparing a cost-effective viral transport medium, you need MEM in powdered form. So that you can use also a beef extract in powdered form. Beef extract in a powdered form you can use. But this is only nutrients. You also need amphotericin B, contamination, and also an appropriate buffer system for maintaining the pH. How to understand the result is false negative. Okay, on the third day, Dr. Irfan will very nicely explain you in detail what is false positive, what is false negative and everything. So you will learn it. This is not simple. So I cannot answer you in 2 lines. Okay. So there are questions. I'm wondering about the minimum requirements, facilities, number of rooms, labs to test for COVID-19 in the border. Well, Haider Hamza, this is not as something very straight. What you need actually, you need an isolated area for RNA extraction. You need a reasonable isolation between the PCR steps. This is what you need. So in a very small scale, if you have an area where you can fit 4 rooms, I think would be fit. But the most sensitive area is the area where you are actually extracting the RNA. That must be a BSL-2 plus safety level. Okay. Now, okay, there is actually a correction or a comment from Robin Abu Ghazala. Thank you very much for your correction. WHO now recommends 10 days plus 3 symptoms-free days for release of patients as a non-infectious virus may be detected by PCR. Okay, thank you very much for all of you for the participation. And there is an announcement at the end again, because you will receive a certificate for this training session. So please mention your correct names in the comment section. All of you mention your correct names in the comment section so that we can prepare correct certificates for you. And once again, I am really thankful to all of you. There is huge participation, a very good number of participants. I am very happy to be with you, and I think this training may help you to some extent, and hope to see you again tomorrow and day after. Thank you so much. Allah Hafiz.